Okay, welcome back, everyone, and let's get started on this second session for the Computational Biology Symposium. So it's a great pleasure for me to introduce Uri Alon from the Weizmann Institute, who will be talking about design principles of hormone circuits.
Thank you. It's a pleasure to be here and talk about hormone circuits. Are fascinating, also very important. For example, the hormone insulin is involved in diabetes, which is an epidemic. Almost 10% of the world population. 1 out of 7 healthcare dollars is spent on it. Where high blood glucose damages the blood vessels, causing lots of damage in the body. And then there's also mysterious diseases like type 1 diabetes, where the body attacks our own cells. It's an autoimmune disease and specifically kills the cells that make insulin. They're called beta cells. And that's a huge evolutionary cost if you think now it's 100 years to the synthesis of insulin. Before that, 1% of children would mysteriously die from a disease called type 1 diabetes. And now they can live because of insulin injections. So why are there autoimmune diseases like this? And why do they attack beta cells and not other cells? These kind of questions I want to address in my talk. So we'll approach complex circuits with simple, they're so complex, we'll approach them with a very simple approach, circuit approach. And I'll use beta cells and glucose as a model system. So blood glucose is kept really constant among different people, 5 millimolar concentration. Too low, below 3 millimolar, and you pass out. You don't have enough glucose. And too high, above 7, starts damaging your body and causing prediabetes and then diabetes. And if you drink some glucose, glucose goes up and down in a couple of hours, and the entire dynamics is very conserved, so that if you have a little bit more glucose in 2 hours, that's a criteria for type 2 diabetes. So this baseline and also the dynamics are very much preserved, constant among people. And the way this works is by insulin, where glucose in the blood can only go into fat cells when insulin binds a receptor in muscle cells. So the transporter is moved to the membrane, and then glucose is removed from the blood, and that's how insulin helps remove glucose, you can say. And insulin is made by beta cells in the pancreas. There's about 1 million islets like this. Each one has a few thousand beta cells. And the feedback circuit looks like this. Glucose is made by the liver when we're asleep, or by a meal when it comes from the gut. And it activates beta cells to secrete insulin that helps remove glucose from the blood within hours. And this feedback that's what you learn in medical school, and it can recapitulate this glucose tolerance test where you drink glucose. There's a parameter here called insulin sensitivity. So how effective each unit of insulin is called insulin sensitivity. And a lot of people have insulin resistance, where this parameter is very low. Insulin is not effective, especially in obesity, due to all kinds of factors. There's insulin resistance also in inflammation, and it's a really great physiological knob. For example, if you have inflammation, you do want more glucose in the blood to help the immune system fight the pathogen. But if it's chronic, it causes problems, including diabetes. And if you change, like in any model, if you change a parameter, like how effective insulin is removed less. So tenfold insulin resistance, you have twice glucose, like 10 millimolar, and it's removed much less. The problem is that most people with obesity have normal glucose, even though they have high insulin resistance. They all have high insulin resistance, but almost all of them have high glucose, normal glucose. So there's something else that has to compensate for insulin resistance. And that extra circuit is another feedback loop, which is that the functional capacity of beta cells, which means how much total insulin they can secrete, increases when you have insulin resistance and obesity. And that's carried out by a feedback loop where glucose actually makes beta cell mass grow, and also beta cell secretion capacity. That's what we call total functional mass. So we have to add to the circuit an extra step where glucose not only makes beta cells secrete, it also increases their functional mass. It's like on the fast time scale, they secrete more insulin, but on the time scale of weeks, the actual mass grows. And that's an interesting circuit, especially seeing that when cells divide and grow, it's a very unstable thing, because if you think about proteins inside a cell, they have production and removal, and that's very stable. You reach kind of a steady state that's production divided by removal wherever you start. But cells live on a knife's edge. Since they divide, all cells come from cells, their number can grow exponentially. So if proliferation is larger than removal, you have an exponential growth, like in cancer. And if proliferation is less than removal, you have a crash, like in degenerative diseases. So just to keep the number of beta cells constant or in a good range, you need a feedback loop that balances proliferation and removal. And that's right, that's the same for every organ. If you think about it, in every second, 1 million cells in our body divide and 1 million die. If those numbers were a little bit different, we would grow or shrink. So every organ has its own feedback loop to keep proliferation and removal matched, which is called the problem of organ size control. And that's what glucose does to beta cells. And this is pretty incredible. Glucose, if you take experiments, this is on rodent islands, and you see how glucose affects, let's say, the survival of beta cells, you can see that beta cells die when glucose is too low, below 5 millimolar, and then they stabilize, and then they start dying again. So it's kind of a U-shaped death curve. Glucose, in low levels, beta cells are killed, then they survive, and at high levels, glucose is toxic to the very cells that remove it, glucotoxicity. So we're gonna consider glucotoxicity later. Just want to walk you through what happens with this removal rate that drops with glucose. Removal rate drops with glucose, and now we can plot also proliferation rate that rises with glucose. And when proliferation equals removal, that's the fixed point. And this is how beta cells know what size to be. So for instance, if you have a little bit more than 5 millimolar glucose, there's more proliferation than removal. Beta cell functional mass grows. There's more insulin, therefore less glucose, and glucose flows down back to 5 millimolar. So it's a stable fixed point. If you have too little glucose over weeks, removal is higher than proliferation. Beta cell mass shrinks, there's less insulin, more glucose, we flow back to the fixed point. And so this beta cells basically change their size until you reach 5 millimolar. And that's a great way to compensate for insulin resistance, for instance. If you have insulin resistance, too much glucose, beta cells grow until glucose goes back to 5 millimolar. And actually, this compensates everything, the dynamics of glucose after meals. The only thing you see that insulin is higher because there's more beta cells. You have more insulin in people with obesity, but their glucose is fine. And so this circuit is amazing. It both keeps size control and compensates for parameters like insulin resistance. And that's maybe why it's found in almost all endocrine glands, like the thyroid gland secretes thyroid hormone under control of TSH. TSH also makes thyroid gland grow. And adrenal has its own feedback loop, and stomach cells secrete acids do. So this circuit, I think, controls organ size and also compensates for parameters across endocrinology. So that's our first principle. But then now let's return to this. Why does glucose start killing beta cells at high concentrations? That doesn't make much sense. And I just want to show you that it's actually horrible for us, because now death and proliferation cross twice at 5 millimolar and at a higher level. And this is an unstable fixed point. If glucose is higher than this, let's say 10 millimolar or whatever, for enough time, it's a vicious cycle. There's more removal than proliferation. Beta cells crash. There's less insulin, and therefore even more glucose, and therefore more killing, and therefore more glucose, and more killing, and more glucose. And it's a vicious cycle that looks a lot like end-stage type 2 diabetes, where you eventually lose all your beta cells, and you need insulin in order to survive. And so this glucotoxicity is really linked with end-stage type 2 diabetes. There's a route to diabetes, which is chronic insulin resistance, and then beta cells kind of compensate, but when insulin resistance hits a threshold, they crash. So it's kind of a limiting a carrying capacity. This all explains different things, like age. Why does diabetes increase with age? There's many reasons, including weight gain, but proliferation of all cells in the body decreases with age. And when proliferation decreases, you see this unstable fixed point move to lower levels of glucose. It becomes easier and easier to cross into this unstable range. and genetic background, we need to know more about this glucotoxicity to understand propensity for diabetes. This is data from Eran Segal, who's speaking next, showing this how glucose rises kind of gradually in prediabetes, with insulin resistance, sensitivity goes down, insulin resistance, and then jumps up at a threshold, which is a kind of prediabetes to diabetes transition. But then we can ask, why do we have glucotoxicity? Why do beta cells die when there's high glucose? Why does this thing they're supposed to control actually kill the cell that's controlling it? I mean, we know how it happens. It happens because of the glucose that makes a lot of glycolysis, and there's reactive oxygen species, which kills beta cells because they're actually exquisitely sensitive to reactive oxygen species. That's the how, but why does evolution make them so sensitive? And we want to propose a physiological role for this glucotoxicity. This is work of Omer Karein. And that is because this circuit is made of cells. It's not like a thermostat that an engineer can build. It's controlled temperature. This is a thermostat that divides and mutates. That's cells. So since these cells divide and mutate, eventually there'll be a mutant that misreads glucose. And that mutant will do what it's supposed to be. This way it thinks 5 millimolar is actually 25 millimolar. It'll say, oh, we need to divide and secrete more insulin. And that's what it'll do. And that'll cause a clone of cells that secrete too much insulin. And eventually, this clone of cells will take over the pancreas. This happens also in the thyroid, called thyroid toxic nodules that think there's too much TSH and secrete too much thyroid hormone. So these mutants are known, mutants in glucokinase, etc. And they're a terrible problem because if you have too much insulin, you have low glucose, and low glucose is lethal. So these mutants can be lethal. They grow and secrete. And so if we didn't have glucotoxicity, this mutant, which thinks that 5 millimolar is 25 millimolar, will proliferate, as I said. Glucotoxicity, making too much insulin and killing us. Glucotoxicity can offer a cell autonomous route to get rid of those mutants. If a cell thinks it's 25 millimolar, it kills itself because of glucotoxicity. It's a cell autonomous thing. And that's a physiological reason why you have a U-shaped response curve. To kill the mutants who think there's too much signal. And experiments where you put in chimeric mice, those mutants, small amounts, show that they really do disappear, but they don't disappear if they're the majority. So this U-shaped biphasic curve happens in other places too in biology. In neurons, like excitotoxicity, and immune cells where they see too much antigen, they kill themselves. And we think it's a general way to protect the body against mutants. But if you look carefully, you see that there's still a range of mutants. Let's say they think that 5 millimolar is actually 8 millimolars, let's say glucose, and they will grow more than die. Black line is above the proliferation line, black line is above the red line, the removal line. So those mutants are still dangerous. So we got rid of the strong mutants. What about these mild mutants? And thinking about those mild mutants, they can still, grow and secrete too much insulin and kill us. Think about those mild. So far, we've seen this organ size control principle where glucose makes beta cells grow. That's a great compensation mechanism, but it makes you prone to these mutants that sense too much glucose. So you have biphasic mechanism to get rid of the mutants, but that reveals this fragility to these mild mutants. And that got us to thinking about autoimmune diseases. And this is work of Jan Cohen and Neil Friedman. And I want to dedicate the talk to Neil Friedman, who passed away this year. He's a pioneering systems immunologist and really gentle and wonderful human being. And so, again, to remind you, type 1 diabetes T cells, for some reason, kill specifically beta cells, and they kill all the beta cells. You don't have insulin, and you need insulin in order to survive. Usually, it's thought that these T cells are a mistake. They somehow bypass the thymus selection and all kinds of selections, and they're a mistake. But we want to, again, ask, maybe they're part of a physiological process. And in fact, to get rid of those mutants, to get rid of these mutants, we reason that there are T cells who specialize in sensing beta cells that make more insulin than their neighbors, according to peptides in the secretion, in the insulin production and secretion pathway. And then they differentially kill those cells. So the T cells are like police that detect hypersensing, hypersecreting mutants and kill them before they can take over the tissue. That's the hypothesis, these 007 cells. This is the paper where you can read about this hypothesis. And that gives a selective disadvantage to those mutants. And if you calculate, by the time you have 10 to the 9 beta cells, and you calculate the mutation rate just to make those 10 to the 9 cells, you reason that you should have thousands of those mutant clones expanding in your body. Also in the thyroid, you have tens of thousands of toxic nodules. And we think you can't have a hormone system without a kind of surveillance. We call it autoimmune surveillance of hypersecreting mutants. And it requires T cells to detect antigens in the pathway making the secreted hormone. And how the T cells must differentially tell the difference between cells that make more or less of those peptides. If there's too little surveillance, you get clonal expansion and all these hyper-secreting diseases. If there's too much, and for reasons we don't understand, basically what we're saying is those T cells are the basic material that, for some reason, in some people, breaks down into autoimmunity, and that, like antibodies and stuff, and you kill the whole organ. And indeed, you have autoimmune diseases in all these tissues that have this circuit, like Hashimoto's in the thyroid, vitiligo for melanocytes, PBC for cholangiocytes, Addison's for the adrenal. This organ, the circuit for size control and mutant resistance appears in all those glands. And in all those glands, the antigens are in the secretion pathway. Like in the thyroid, there's 2 enzymes at the end of the making thyroid hormone. And they're the famous TG and TPO antibodies that about 5% of people in the population have thyroiditis, mostly women. And those are the auto antigens. And in all these diseases, the autoantigens are in the secretion pathway. They're hydroxylase for cortisol, etc. So that explains why those peptides and not others for autoimmune diseases. These are all these autoantigens. And T cells can also work this way. They can, I mean, if the T cells are very strong to detect an antigen, they needed basically one antigen present another cell to kill it. But if it's a intermediate recognition, they can tell the difference between a cell that's making 10 or 100 presenting peptides. And that's why T cells can do this task. And this explains also why everyone has T cells against beta cells, T cells against thyroid. They exist in all healthy people. And this explains they have, we think, a physiological role to protect against these mutants. And this also kind of addresses the question of why some cells are attacked and others aren't. For instance, thyroid and beta cells and adrenal cells are attacked by the body. But then there's parathyroid gland, a tiny gland on top of the thyroid, that never gets autoimmune diseases except for rare mutations or checkpoint inhibitors. The alpha cells near the beta cells that make glucagon never get an autoimmune disease, or renal cells that make renin don't. So what's going on? Why do some endocrine hormone organs have autoimmune diseases and others don't? And we say that according to our theory, those organs don't have this checkpoint, this immune cells killing mutants. And so they're prone to actually these mutant expansion diseases. And that actually is true. Parathyroid gland is a very common disease where adenomas make too much parathyroid hormone and you get too much calcium, etc. So these predictions tell you a trade-off between 2 kinds of diseases, autoimmune diseases and diseases of mutant expansion. And we think that organs where it's very bad to lose the hormone don't get autoimmune diseases, whereas organs where it's very bad to have too much of the hormone, like insulin, you have too little glucose in your diet, they get autoimmune disease. So it's kind of a trade-off between 2 kinds of the diseases. So just to summarize, actually, I made a musical summary. I'll just say, we walked through showing that you need organ size control to stabilize organs, and that requires a circuit where the signal kind of controls cell proliferation to keep organ size control. But that makes you very sensitive to cells that missense the signal. And therefore, you can have these biphasic curves which kill cells that sense too much. But that opens up this range of mild mutants, and that means you need this something like a T cell removal, but that gives you the material for autoimmune diseases, right? So it's all basic biological principles that can explain a lot about diseases. I'm just a T cell and my story's seldom told, educated in the thymus. I learned how to defend my country, laying low, seeking out those lymph nodes, where the other T cells go, seeking out those places only they will know. When I first came to the pancreas, I saw a lot of beta cells secreting insulin. Then I saw a hypersecreting mutant, making so much pre-pro-insulin, it threatened to become an over-secreting clone. I pulled out my dagger, did what a T cell has to do. 99, 99, 99, 99, 99, 99. Oh, I hope those T cell, beta cell clones don't expand. Make me call in the B cell army. Start a civil war with brother cell, killing brother cell. This we must avoid, after all, I am a pacifist. 99, 99, 99, 99, 99. So I'd love to get any questions and learn from you. Thank you very much.
Thank you very much, Uri, for that fantastic talk and the final song and guitar play. Really lifted the whole meeting, I have to say. So I start from with a question from an anonymous attendee here, which is wondering how does T cells detect which cells are secreting more? Do you have an idea what the sensory mechanism is there?
Yeah, in our theory, we think that, first of all, there's a mean field thing, which is regulatory T cells sense the average level, and so they inhibit. And then the cytotoxic T cells, they go around the cells, and they can tell which cell goes above that mean field, because they can detect differential levels. So let's say they see cells making 10 or 20 MHC class 1s with that peptide, and suddenly they see a cell with 100. And that gets them primed. So the entire organ can secrete more. The Tregs, regulatory T cells, look at the mean field. But if there's an outlier cell locally, then T cells can recognize it and kill it. That's the proposed mechanism. I hope I answered your question.
Thank you very much. Yeah, so an anonymous person can follow up if they needed more information there. So a question from Scott Tyler, who also enjoyed the song at the end there. So the question is, the T cell check on proliferation model is interesting. Have you looked for something like lower expression of PD-L1 or similar on these types of cells that have a T cell feedback loop?
Scott, that's really interesting. No, we haven't, and I guess we can compare organs like parathyroid and thyroid in terms of PD-1, if I understand you correctly. And if not, just please write some more and give me more context. But we haven't done that.
And another question from an anonymous person here is there a connection between the risk of autoimmune disease and the risk of cancer in the same tissue?
Yeah, there's a very interesting connection. Like, for example, so the risk of cancer generally rises with the size of the tissue and the number of stem cell divisions. The skin is a huge organ with a lot of stem cell divisions, so is the gut. That's the risk of cancer. But you can still see correlations. Like, for example, people with the autoimmune disease that attacks melanocytes do better when they have melanoma, which is a cancer of melanocytes. And also thyroid cancers, which are quite deadly, they are very interesting. They don't secrete thyroid hormone. They're what's called cold in terms of thyroid hormone. Whereas toxic thyroid nodules, which secrete thyroid hormones, are adenomas and they don't become cancerous. So it looks to us like to become cancerous, one step that the cancer evolves is to shut off making the hormone, and maybe but that way evade this whole surveillance system. And that's what actually makes those cancers so deadly. If they did secrete thyroid hormone, you could see it because you get hyperthyroidism, you can catch them. But since they don't, you only catch them at a very late stage. And so there's definitely a link that we'd love to explore more between cancer and autoimmune diseases. I hope I answered the question.
Thank you very much. And one more question from an anonymous person here. So he's wondering, are secretory antigens specifically regulated differently than other autoantigens by air to allow for the development of these autoreactive cells?
So anonymous attendee, I really think this question is extremely perceptive, and this is one mechanism we'd love to follow up, and that is that it's possible that the thymus, which trains T cells, has a secret agent training school. That is, it has a place or a way to specifically express those antigens that are useful for destroying mutants, like pre-proinsulin or TG and TP, all these famous antigens, and prepare special T cells that have this task. For example, these T cells might not need a second signal to tell them it's a viral attack. Maybe they have a license to kill. And that's very interesting, because then those T cells might have a handle that we can use to control autoimmune diseases. And I don't know if anybody knows. I'd love to know if those autoantigens that are specific to autoimmune diseases are differently regulated by the thymus in this respect you mentioned, air. I think that's a great hypothesis.
Thank you very much. So I think we ended very much on time here. So I'd like to thank Uri again for the great talk and for the great song. And we will switch over to Eran Segal, who's